Hi everyone. Now that we've learned about how we determined that DNA was indeed the genetic material, all right, from Griffith's experiments, Avery, McLeod, and McCarthy, and then also Hershey and Chase, this is what we worked on last Friday. Now we want to know how DNA replication occurs, and that's really what this week is going to be all about. So we're still on Chapter 9, DNA replication. The key objectives for this particular video, at the end of this I really want you to understand the requirements for cell division and why DNA replication is kind of part of that. I want you to understand the structure of DNA, so we'll review that once again, including the DNA helix and also the anti-parallel strands and base pairing. All these things that we learned in Chapter 3 we're really going to emphasize now because this is important for the last several chapters of this semester. And so we'll also talk about how that structure was identified and the people involved in that. I want you to understand the basics of DNA replication and semi-conservative replication. We will go into details of this over the Wednesday and Friday together. But the basics of this is really important so that when we do go over those things together, you're ready to understand. And we will talk about Mieselson and Stahl's experiment that demonstrated semi-conservative replication. And so this is important here because I want you to be able to identify and predict the results that would have occurred for various methods of replication. So this is part of thinking like a scientist. It's not necessarily Mieselson and Stahl's experiments per se that I think are extremely important, although they were definitely groundbreaking and hugely important for our understanding of DNA. But this is part of our goal to really teach you to think like a scientist. So some of these experiments are difficult and the analysis can be difficult, and we're going to work on that together in this video. And so hopefully by the end you should be able to understand what their experiments were and how they demonstrated, how the results that is demonstrated that it was semi-conservative replication. That is how our DNA replicates itself. And with all that, that's going to allow you to be ready for our synchronous discussions on Wednesday and Friday on the mechanisms of bidirectional synthesis. So that's where we're headed here. All right, so as we start talking about the structure of DNA, it's really important to mention Rosalind Franklin. So Rosalind Franklin was a PhD student in Dr. Wilkins' lab, and she used X-ray crystallography to study isolated DNA molecules. So I certainly don't want you to go into the physics of this. It's pretty interesting, but can be difficult. But if you shine X-rays through a structure, those X-rays will diffract depending on what it hits. So even molecules obviously have density, and the atoms are going to have density, and where those atoms are is going to kind of lead to a pattern, a diffraction pattern. And so computers and knowledgeable people can deconvolute this and really figure out what the structure is based on the diffraction pattern. So this is actually how we learned about the three-dimensional structure of proteins that we've talked about in the past. This is also how they learned about the three-dimensional structure of DNA, which we know to be the DNA helix. However, Rosalind Franklin's name really got lost in the shuffle, and you probably hear about Watson and Crick. They're given the credit for discovering the structure of DNA, and they actually did quite a bit to really progress the understanding of the DNA double helix, but really it was Rosalind Franklin who was the first to identify the structure based on this X-ray crystallography. And so somehow Watson and Crick obtained that critical X-ray data, and both of them actually have recently acknowledged that it was her image, her data, that inspired their model of DNA. Unfortunately, Wilkins didn't really advocate for Franklin. He probably was a bit of a sexist, and in her being a female, especially at that time, really didn't get the credit that she deserved. And so I think it's really important that we understand Rosalind Franklin's name. And I've been pretty impressed lately. Every year prior to this, each year people seem to know Rosalind Franklin's name more and more before they even come to class. So hopefully her name is getting out there in the mainstream in high schools and so forth as well. But her data was really, really important. Unfortunately, her paper was published third, still in Nature, so it was a great paper, but it wasn't given the due credit, and the first and second paper by Watson and Crick are the ones that really got the credit along the way, even though her data was critical for all of this. Sadly, she died in 1958, way too young, of ovarian cancer, probably due to her work with these X-rays, because at that time people didn't realize how dangerous X-rays, X-rays and that radiation could be. All right, so let's talk about the structure of DNA. So this is the double helix, and this is actually just a picture of the model that I would normally bring to class. And so with this structure of double helix, we basically have the phosphate backbone. So the phosphate backbone links the DNA together, and then we have two of those wrapping around each other. So this is one of those strands right here, and then this is the other one, and they kind of wrap together, the double helix, and the bases stick in the middle, and those bases are linked together by hydrogen bonds. And so this is kind of like a spiral staircase. So the phosphate backbone, these are covalent bonds, very strong, so these are not going to break within that strand. So this is like the handrails of the ladder, or the kind of the sides of our spiral staircase. And then the hydrogen bonds between the two strands, those are weaker, so they can unzip, and this is going to be important for DNA replication. So you can break those double helix, the hydrogen bonds holding the double helix together, and separate the two strands, and that's the first part of replication, actually. So the DNA bases held together by these hydrogen bonds, that's kind of like the steps of our staircase. So the X-ray crystallography model really suggested two helices. So the only way to explain the data they got was that there had to be two helices linked together, and the distance between this phosphate backbone, the part that had some density here, showed that it had to be a purine and a pyrimidine, right? So we had to have one of our double ring bases, either A or G, always bound to one of the single ring bases. And the reason for that is if it was a pyrimidine, two of the single ring bases, then it actually was too thin. So the distance between the phosphate backbones, the phosphodiester backbones, were too far apart to be satisfied by this. And purine would have kept those a little bit further apart, and so the data showed that it was closer than this, and so the data fit perfectly with a model where you have a purine and a pyrimidine bound together at all times. And we know that to be the case because we know A always binds to T, and in fact we learned that A always binds to T with 2 hydrogen bonds, and we know that G always binds to C with 3 hydrogen bonds. And A and G are our purines, and T and C are our pyrimidines. Right, so that's always the case, and that DNA double helix basically stays an a consistent distance apart. So we also know about DNA's directionality. Right? So briefly, in the question box, describe why we have a 5' end and why we have a 3' end and how that is named. Well, we know the 5' end and 3' end are based on the carbons of our ribose sugar. So here is our base, all right? This base happens to be thymine. This is our ribose sugar, and this is our phosphate. And the phosphate, ribose, that's the phosphodiester linkage. That's the backbone with the bases sticking in between. So the phosphate group of the 5' end of one of these nucleotides binds to the 3' carbon of the next one, and that just keeps going together. But we know the 5' and 3' end is based on the numbering of the carbons in ribose. And we learned that you number from the right. So there's carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. So this would be the 5' end of that nucleotide, and this would be the 3' end of that nucleotide because it's where the 3' carbon is. This is where the 5' carbon is. And as I said, then they just link together. So the phosphate group, here we have the 5' to 3', and that 3' links to the phosphate on the 5' carbon to 3'. So they're always linking in that same order, 5 to 3, 5 to 3, 5 to 3. So this whole strand also has directionality. We have the 5' end and the 3' end down here. And then when the two strands link together, they're anti-parallel. So one strand will be in the 5' to 3' direction, right? So here's our 5' carbon. There's the 3' carbon, 5 to 3, 5 to 3. So we have 5, the whole strand has the 5' and 3' ends. And then the other one's the opposite. So this is our 5' carbon now, right? So this is the oxygen, carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. This is the 5' end. There's the 3' end. So this is 5' at the bottom, 3' at the top. Anti-parallel. This one's 5 to 3, top to bottom. This one then is 3 to 5, top to bottom. All right? And again, here we have our DNA bases sticking in the middle, creating those hydrogen bonds that links the double helix together.